We're going to be labeling your model of transcription and translation, and what we're going to focus on right now is actually just translation. And this model can be found in your packet for Power Up Your Protein Escape Room. It should be on the 3rd page of it. So we have this single strand here first with nucleotides on it. This is mRNA. And then that mRNA, so that's what got transcribed from the DNA in the nucleus. The mRNA leaves the nucleus, goes out into the cytoplasm, and it attaches to a ribosome. The ribosome is this kind of little blue blobby thing here that is kind of in the background and the foreground. Then on the mRNA, every 3 mRNA bases is a codon, and every codon is going to code for a specific amino acid. Now, the way the amino acid gets brought in is it comes in on a tRNA. The tRNAs are these little T-shaped things that to me look like they have a foot on them. On the bottom of the tRNA is what we call the anticodon. The anticodon is going to be kind of you can think of it as like it's the opposite bases of the mRNA. So for example, here on the mRNA where we have an A, the anticodon on the tRNA is going to have a U. Or like here where it has a G, it would have a C. So basically, the anticodon is what recognizes the codon and it hooks that tRNA up. That tRNA with that anticodon has the specific amino acid that the codon is coding for. So on the top of the tRNA is the amino acid. Then, when the tRNA comes in, it's going to release the amino acid onto the growing chain of amino acids. We call a chain of amino acids a polypeptide. And you can remember that because amino acids are held together in the chain by peptide bonds. So as amino acids get hooked on and we connect them together by peptide bonds, we call that a polypeptide. So like many peptides you can think of in that word. The difference between a polypeptide and a protein is that a protein actually is a fold polypeptide. The folding of the protein is what actually allows the protein to do its job.